Congratulations on winning the travel award from us, and it's again a wonderful paper. Really cool to break open this field again of zebrafish biology. I was just wondering, we were kind of talking earlier about when you showed the differences between the male and female physiology. I was wondering if you could kind of recap that for us again, because I found that part really fascinating.
It's first of all, I'm really grateful to VisualSonics and FUJIFILM for the travel award. It really means a lot to us young, early career cardiology researchers to be able to get some funding to travel to international awards. I'm from Sydney, Australia, so it's a long flight. It's a long flight to come to Los Angeles and the United States, but I'm really happy to be here, and so thank you.
Just the recapping kind of the differences that you showed between the male and female zebrafish physiology, because I think that really speaks to careful design of experiment. I mean, there's so many. Historically, if we look back at some of the zebrafish work that was done, and even some of the mouse work and rat work that's been done in the past, there hasn't been as much of a focus on, age match, cage match, litter match, gender match animals, and especially with zebrafish, as you were saying when we were talking about it, people historically just kind of scooped out all the fish out of the tank and just image or tested or whatever with no regard to the gender. And I thought it was really powerful what you were saying about the gender differences that you showed.
So a couple of years ago, when we first started to work on the adult model of zebrafish as a valid cardiovascular model system, we made some observations that, first of all, the male and female fish were very different in terms of appearance and size, and the females were generally at least 1.5 times heavier than the males. And classically, from physiology, if heart size is associated with body weight, we would expect differences in terms of heart size and structure, and potentially even stiffness or diastolic function. And so, based on this, we decided to segregate all the animals, we gender segregated them first, and then we followed them with time. And what we found was that it was true that the males and the females were very different in terms of their size and structure, but particularly the diastolic function. The females were definitely a lot more stiffer for the same age relative to the males. And when we did that, we realized that for future experiments, we shouldn't group these animals together, and we should actually match according to age and gender. And so our initial studies for the first couple of years were based on working out biology of the zebrafish, working out how to best replicate results or produce results that were reproducible. So, as we all know, not just biological variation, but also there are operator and technical factors which impact on reproducibility. So what we did was we carefully designed studies to work out exactly what caused biological variation. For instance, anesthesia is a big issue, and we worked out the effects of anesthesia on heart rate. What we actually did was we then tried reverse translating all the usual clinical echocardiography principles, such as strain rate, which is actually possible within the limits of the Vevo 2100 resolution. And we were able to do diastolic strain rate in the zebrafish. In the work that we presented in the American Heart Association in the conference, what we showed was we were actually using the zebrafish adult model system to recapitulate the disease phenotype. So basically, there's a mutation in humans known as a titin truncating mutation, which is associated with dilated cardiomyopathy. My second lab at the Chang Research Institute, and a senior postdoc there, Dr. Inken Martin, managed to create an exact homologous model of this human A-band truncating variant, where they knocked out, or they caused a truncated mutation in the A-band of the titin. And we studied the fish. In humans, the mutation is associated with dilated cardiomyopathy. And we noticed that a few family members who carried this mutation, if they were subjected to volume overload, that their cardiac function deteriorated quite rapidly.
And that's consistent with family genetics. There's clearly a connection. If you go back with the
so in the genetic analysis, what happened was in two separate families, individuals who had chronic volume overload developed severe cardiac dysfunction, and quite rapid dysfunction. And so we could reproduce the effects of volume overload in a different animal model system. So we did it in the zebrafish, and what was amazing was that was true. That volume overload had precipitated severe heart failure and cardiac dysfunction, both in systole and diastole. So there were diastolic effects too in the zebrafish. And this sort of means, and why I think sometimes the zebrafish model is actually quite powerful, is that if you have conservation of a mechanism or a disease mechanism despite 400 million years of vertebrate evolution, that's a pretty damning mechanism. I mean, that's conserved despite 400 million years, and that's what we presented today. And that work, all of this work in zebrafish, the insights into understanding how the animal is or how it lives, both in health and disease, is not possible without high-frequency echocardiography. And that's where we're very grateful. I mean,
that actually is perfect, because that leads to my next question I was going to ask you, because you had mentioned earlier about not only being able to do echo in the animals, but also being able to do the strain analysis and, you know, recapitulating all of these clinical tools and clinical approaches. So I was going to ask you what the Vevo imaging technology has meant to you in your research, what it's been able, the kinds of doors that it's been able to open for you in this type of research.
Oh yeah, I mean, the Vevo is just amazing. To get to 30 to 70 megahertz allows us to do all sorts of things, not just looking at the systole, but particularly diastole. And I guess this is where high-frequency echocardiography from Vevo and the zebrafish, you know, it's almost like a perfect union, because high-frequency echocardiography has some limitations. For instance, depth of penetration is a bit of an issue, but with the zebrafish, depth of penetration is not an issue, because the animal is only 1 centimeter deep, like in terms of its maximum height. And so we do not have issues with frequency, dependent attenuation. The fact that we can actually tease out diastole in all phases of diastole, particularly because of the Vevo technique, it's just so eye-opening. People in the field of transgenic cardiomyopathies and animal models have, until recently, only focused on what's happening in systole.
Yes, and that's been the trend that I've noticed too.
And diastole, I mean, just broadly speaking, is at least 2/3 of the cardiac cycle, and it is a very important part of the 2/3 of the cardiac cycle. And so now your technology is enabling us to have a look.
That's interesting, because that's a question that we get from time to time about diastolic analysis. There's kind of two trends that I've noticed that have popped up from time to time more often as we go forward, and that's right ventricular functional analysis. For years and years, it seemed that people kind of ignored the right side and just focused on the left. So now there's been a lot more inquiries about right ventricular analysis, pulmonary hypertension assessment, and the other one is assessing diastole. How do I assess diastolic dysfunction, because systolic function has really again been the focus for so long. So that's interesting to hear that it really contributes to being able to do that with technology.